Hello. First of all, I want to thank the organizers to invite me and participate in this nice symposium that is always very nicely organized, and it's a really great pleasure for me. And I'm going to talk about the enzyme recognition of substrates and inhibitors using molecular modeling tools. Here is the simple scheme of the interactions between the enzyme and substrate. And actually, what we are interested in is enzyme substrate complexes. So these complexes are formed if the enzyme and substrate fit each other with both respect of electrostatic and steric features. And after that, the chemical reaction takes place. And what is interesting is that enzymes can efficiently perform chemical reactions that cannot happen in solutions. So therefore, some specific things happen in the active site of enzymes, and we want to somehow quantify and demonstrate this. Actually, the question that we are going to answer is what is the substrate activation in the active site of enzyme, whether we can quantify this or even, well, or at least demonstrate. So what we use as molecular modeling tools is a combined quantum mechanics, molecular mechanics approach. This is, so actually it was already discussed today in the previous talks. And what I want to add here is that we shift from the study of local minima corresponding to the enzyme substrate complexes to the ensemble of states. This can be done if we utilize molecular dynamics simulation with the combined quantum mechanics, molecular mechanics potentials. And as a result of this simulation, we obtain the set of MD frames, so at an MD trajectory. And we can analyze it with from different points. Well, of course, the simplest way is just to analyze geometry parameters, like, for example, interatomic distances that we supposed to be important in the particular case. Or as we perform QMMM simulations, at each MD step, we can, we obtain the wave function, and we can calculate electron density and descriptors that are based on this electron density, and also obtain some distributions of these values. So therefore, we move from the single structure to the set of structure with the distributed parameters. So here is a short advertisement of the software that we utilize. It's a GPU-based DFT code called TerraChem that is really fast and extremely useful for our applications, as we can utilize GPU cards just gaming cards, and rapidly calculate energies and forces in QM subsystem during QMMM simulations. So it's like 1 or 2 minutes for each step, energy and energy gradient calculation. So let's move to the examples. We are interested in the enzymatic reactions that are initiated by the nucleophilic attack of the carbonyl group. So here is the general picture of the active site. The substrate has a carbonyl group. There should be a nucleophile, and always there is an oxyanion hole that polarizes carbonyl group and prepare it for the chemical reaction. So actually, nucleophilic attack is widely found in enzymatic reactions. Those are hydrolases of different types that can hydrolyze peptide bonds, other CN bonds, ester bonds, or carbon-carbon bonds. And nucleophiles are also very different. Those can be the side chains of amino acid residues, or in case of metal enzymes, those are water or OH minus anion. So we'll focus on these 3 examples marked here. And the first one is the bacterial enzyme metallo-beta-lactamase. This enzyme is responsible for drug resistance. So what it does, it hydrolyzes the beta-lactam containing antibiotics. Here is the example of imipenem. It's the widely used antibiotic that can be in pharmacy. And the chemical reaction is initiated with the nucleophilic attack of the OH minus on the carbonyl carbon atom, and then as a result of the reaction, the CN bond is cleaved, and the hydrolyzed imipenem is not already is deactivated with respect to its antibiotic. So we wanted to somehow demonstrate what happens with the substrate with this imipenem compound upon binding to the active site of the enzyme. We compared the QMMMD trajectories of the imipenem in water solution and in protein. Here in protein, the carbonyl group is coordinated by zinc cation, of course, this cation should somehow polarize carbonyl group. So we can observe it from the distributions of the Foucault electrophilicity indexes that are calculated as shown here. And for imipenem in solution, these indices are small in the entire trajectory, and for protein, the distribution is wide, and there are values with quite high electrophilicity indices. That is an indication that in protein, there is a huge fraction of states with the imipenem being ready for the nucleophilic attack. So here is one example. Another example is also related to the bacterial resistance, and it's another bacterial protein, penicillin binding protein. It's the enzyme that is responsible for the formation of cell wall in bacteria. And actually, antibiotics that contain beta-lactam group, they are bound to these enzymes and block enzymatic activity. So that should kill bacteria. But of course, as the resistant mechanism, resistant strains appear with the mutations in this gene coding PBP2 protein. And actually, PBP2 protein from the mutant strains demonstrate considerably lower specificity towards ceftriaxone. It's another antibiotic. We find the more than 100 time decrease of the specificity constant in one case, and actually even much more in case of HO41 strain. So we wanted to analyze enzyme substrate complexes of PBP2 proteins from these two strains to somehow understand what happened in this case. So here we utilize the simplest criteria that are based on geometry parameters. So this is the catalytic serine residue that initiates the nucleophilic attack. And as geometry parameters, we used the nucleophilic attack distances and also hydrogen bonds that are responsible for the oxygen hole formation. So here you can see these distributions. As for the nucleophilic attack, the distribution is shifted to the smaller values in case of PBP2 from the wild type strain. Still, we cannot discriminate these two mutant strains from each other. So the hydrogen bond is always formed for all three PBP2s between the oxygen atom of the substrate and the H group of the threonine, whereas the situation is very different for the hydrogen bond with the serine residue. So in case of the wild type, the PBP2 from the wild type strain, we can see the narrow distribution centered at around 2 angstrom that correspond to the hydrogen bond, Whereas in case of PVP2s from the mutant strains, these distributions are shifted to the larger values. So another important feature is the geometry criterion is the difference of two interatomic distances of two hydrogen bonds, as it allows to quantify not only the individual parameters but also their interrelation at each frame. So we can find that this distribution corresponding to the PVP from the wild type strain is centered at around 0, indicating that both hydrogen bonds are almost of the same length at each MD frame. And if we move to the larger values, we can conclude that for the less reactive 3502 strain, the distribution is shifted to the larger value, and for the least specific strain, PBP2 from the least specific strain, this distribution is even shifted more to the larger value. So even simple geometry criteria can be utilized to quantify specificity in this particular case. And of course, I cannot skip the important example of the main protease from SARS-CoV-2 virus. We all know that this enzyme is highly specific to the certain type of the amino acid sequences. I mean that for the hydrolysis reaction to take place, it is important to have glutamine in the P1 position right right before the cleaving bond. And also it is important to have a particular residue, amino acid residue at P2 position that is actually located quite far from the cleaving bond. Still, it is important to have lysine in this position, and other substitutions result in the decrease of the KCAT value of the catalytic rate constant. So we wanted to explain this substrate specificity in terms of the substrate activation efficiency, and here is the scheme that represent the following thing, that there are two conformations, reactive and non-reactive ones, and only reactive conformations can initiate the following nucleophilic attack and chemical reaction. So in the first example, we analyzed the Fukui function, but those were continuous distributions, and we wanted to find some criterion, some way to discriminate binary, non-reactive and reactive species. So we searched for such criteria using electron density features, and we find quite an old example of the Laplacian of electron density analysis of some small molecules. It was in the late 80s, and the Laplacian of electron density is an electron density descriptor that is calculated as the sum of the second derivatives of electron density over spatial coordinates, and it allowed to discriminate electron density depletion and concentration areas. In other words, electron density depletions are electrophilic sites, and electron density concentration areas are nucleophilic sites. So we analyzed our systems from this point, and we found Laplacian of electron density maps of two types. So the right one corresponds to the reactive species, and we find here the area of electron density depletion. That is electrophilic site for the chemical reaction. And on the right part of the slide, you can see the non-reactive species. In this case, the carbon atom is surrounded by the electron density concentration region. That, of course, is not favorable for chemical reaction. So, not to analyze Laplacian of electron density at all steps, at all frames, we calculated a set of Laplacian of electron density map and reduced our system to 3 coordinates that is supposed to be important. And we found that if if the state is characterized by small distances of hydrogen bonds in oxygen ion hole and also the distance of the nucleophilic attacks, then the system demonstrates substrate activation from the point of view of Laplacian of electron density. So we obtained distributions over these 3 distances, and here green bars mark reactive species and magenta are for non-reactive. We calculated these distributions for the most reactive substrate containing lysine, for the less reactive with isolysine, and for the least reactive with alanine. So here are the fractions of the active, well, of reactive species along MD trajectory. And we suppose that those are responsible for different kcat values. Therefore, we recalculated kcat values relative to these quantities. And also we normalized experimental kcat values and obtained very good agreement between these 2 columns. So also I will show you a short example found in the literature. It's an important study of the ring opening process of caprolactam and caprolactone. It's a comparative study, and these 2 compounds differ in the X atom. It's either oxygen or an H group. And it's known that caprolactam is not hydrolyzed by this enzyme. And this is the graph from this theoretical study. And luckily, we found the equilibrium geometry configurations corresponding to stationary points of reactants in the supporting materials. And we calculated Laplacian of electron density at these points. What we found is the following. So for the caprolactam that has very high energy barrier of the reaction, we can see no activation in the ES complex. At the same time, for the caprolactone, with the calculated smaller energy barrier, we found independently that the activation is observed and that there is a small area of electron density depletion. So this criterion is quite universal and can be utilized further. So, of course, during MD simulations, we obtain thousands of frames of structures, and of course we can manually divide these structures as reactive or non-reactive, but also we can use modern tools like neural networks and analyze these frames and divide them to reactive and non-reactive that we are doing now. And of course it fastens the process and will make it more sort of up to date. So with this I want to finish. I want to thank scientific collaborations because this study is done in great collaboration between Moscow State University Fundamental Research Center of Biotechnology and Russian University of Chemical and Technology, and of course financial support from our foundations. And thank all of you for your attention.
Thank you very much, Maria, for a brilliant talk. We have 10 minutes for questions, so feel free to ask. So the first question is from Dmitry Shulga. The question is the following: do you use additional means above molecular dynamics to ensure that the slow-moving collective degrees of freedom of enzymes are sampled well. These modes, he means probably slow-moving modes, are believed to be closely related to enzyme activity.
We start with actually to tell more about the computational protocol. Of course, we start with quite a long classical MD trajectory and analyze general behavior of the system. And actually, what is happening in the enzyme substrate complex is very local movements. For example, here we analyze hydrogen bonds that really does not change much during the simulation. So to answer the question, I can say that first we perform a long classical MD run and analyze what type of movements are there. We can use a method like dynamic network analysis and so on. And after this, we concentrate on the small short trajectories with the QMMD potentials of EF.
Okay, thank you. The next question is from Mohammad Sabri. How long is the simulation time was set for the molecular dynamics?
Well, the typical times for classical MD simulations are hundreds of nanoseconds, like 200 or 300 nanoseconds. And for QMMD simulations, it is like tenths of picoseconds. With the 1 femtosecond integration time step.
So it's much smaller than like collective movement of the peptide itself, for example.
Yeah.
And do you think that it's just continuation of Dimitri's question? May it happen that that some collective movement, some twisting, for example, of peptide, will influence this enzymatic activity, and the description of the reaction that you are providing, it will be drastically changed?
Well, of course, it might be so, but I'm not sure that it's the case, because we perform preliminary long MD simulation and we analyze what is happening. We check whether our structure is already equilibrated and so on. Therefore, I'm not sure that some huge reorganization should take place after the formation of the ES complex. So anyway, chemical reactions should be quite rapid.
Okay, thank you. And since I don't see other question, I will ask mine. Is it possible, how do you think, is it possible to use the approach which you are give here as a predictive tool for predictive modeling or predictive like drug design, some in such predictive settings, I mean?
Well, I think it might be used, though it's quite expensive. Therefore, of course, you cannot check many compounds. And also, I think there is always a huge problem with general sort of physical chemical features like solubility, penetration to some cells, and so on and so on. So, of course, from our side, we can take some distinct compound and say whether it's reactive or not. But it's a small step towards real drug design with the experimental evaluation.
Okay, and the second questions I had is, so you analyze the MD trajectory with some descriptors, from structural descriptors or electronic descriptors, but how do you select them? Is it kind of, do you go from some mechanistic point of view, or it's like or you're just try many of them and find better or worse? Is there some place that it could be non-physical descriptors found in this way. I mean that having in mind a lot of possibilities, you select the one that correlates the best, but this correlation is nonsense.
Well, okay. Actually, all these descriptors that I showed are interrelated. I mean that, of course, for efficient substrate activation, the short hydrogen bond distances in the oxyanion hole are required. So it's the things that are related. But I mean that geometry criteria are, well, of course, they're most simple and can be rapidly calculated. Still, they demonstrate sort of they do not demonstrate the origin of effect. So the electron density based descriptors are more, how to say, they can demonstrate what is happening with the system from the electron density point of view. And at this respect, actually, both calculation of electron density and Fukui function demonstrate the same. But well, for Fukui function, well, we get the distribution, and sometimes it's really difficult to just make the to divide all these values to, for example, reactive and non-reactive space. From this point of view, the calculation of electron density is better, as it is a binary, sort of binary descriptor. There is either, well, so either there is an activation or there is not. And but anyway, these three types of system descriptions are consistent with each other. So they just give different extent of utilization or explanation.
Okay, thank you very much. Is there any other question in the audience? So if not, I propose to have a 2 minutes break right now for pouring some coffee for us before we come back here and listen to oral presentations from other participants. Thank you for Maria for the presentation.